Module 1, Foundations of Immunology and Immunochemistry. Introduction. Welcome to the first module of the course titled Immunology and Immunochemistry. This module explores the foundational concept of immunobiology, including the nature of antigens, antibodies, immunity, and the structural and functional aspects of the immune system. By the end of this module, you will understand the major elements that govern immune responses and the biochemical interactions that take place during antigen-antibody reactions. Let us begin by understanding the basics of immunobiology. Basic immunobiology. Immunobiology is the branch of biology concerned with the study of the immune system in all organisms. The immune system plays a vital role in defending the body against disease-causing pathogens such as bacteria, viruses, fungi, and parasites. It consists of a complex network of cells, tissues, and organs that work together to identify and eliminate foreign substances from the body. The immune system is capable of distinguishing between self and non-self. This capacity is essential for effective defense mechanisms, as it allows the body to attack only foreign invaders while avoiding damage to its own cells. Antigens, antibodies, and their reactions. Antigens are substances that the immune system recognizes as foreign. They trigger an immune response and are usually proteins or polysaccharides found on the surface of pathogens. Antibodies, on the other hand, are proteins produced by B cells in response to the presence of antigens. These antibodies bind specifically to the antigens to neutralize or eliminate the threat. The interaction between antigens and antibodies forms the basis of many immunochemical reactions, which are used in both diagnostics and research. These reactions include precipitation, agglutination, and neutralization, among others. Concepts and types of immunity. Immunity is the body's ability to resist or eliminate potentially harmful foreign materials or abnormal cells. It is broadly classified into two types: humoral immunity and cell-mediated immunity. Humoral immunity involves the production of antibodies by B cells. These antibodies circulate in the blood and lymph, binding to antigens and neutralizing them. Cell-mediated immunity, on the other hand, involves T cells that destroy infected or abnormal cells directly. This form of immunity does not involve antibodies but relies on the activation of immune cells. Both forms of immunity work together to provide a comprehensive defense against pathogens. Immunogens, antigens, and haptens. An immunogen is a substance capable of inducing an immune response. While all immunogens are antigens, not all antigens are immunogens. An antigen is any molecule that can be recognized by the immune system, while haptens are small molecules that, on their own, cannot trigger an immune response but can become immunogenic when attached to a larger carrier molecule. Understanding the differences between these molecules is crucial in vaccine development and allergy studies. Classification and structural motifs of immunoglobulins. Immunoglobulins, commonly known as antibodies, are classified into 5 major classes: IgG, IgA, IgM, IgA, and IgD. Each class has a unique structure and function. IgG is the most abundant antibody in the blood and is critical for long-term immunity. IgA is found in mucosal areas such as the respiratory and gastrointestinal tracts. IgM is the first antibody produced in response to an infection. IgA is involved in allergic reactions. IgD is mainly found on immature B cells. The basic structural motif of an antibody consists of 2 heavy chains and 2 light chains arranged in a Y shape. Each antibody has a variable region that binds to specific antigens and a constant region that determines its class and function. Antibody combining sites and structure-function relationship. The part of the antibody that binds to an antigen is called the antigen binding site, or paratope, while the corresponding part of the antigen is called the epitope. The high specificity of antigen-antibody binding is due to the unique shape and chemical properties of these regions. This structure-function relationship is critical for the antibody's ability to recognize and neutralize a wide range of antigens. The specificity also allows antibodies to be used in various diagnostic and therapeutic applications. Complement system and fixation. The complement system is a group of proteins that enhances the ability of antibodies and phagocytic cells to clear microbes and damaged cells. It promotes inflammation and attacks the pathogen's plasma membrane. Complement fixation refers to the activation of the complement system following antigen-antibody interactions. This process leads to the formation of membrane attack complexes that can directly lyse the cells of invading microorganisms. Organization of immunoglobulin genes. The diversity of antibodies is made possible through the unique organization of immunoglobulin genes. These genes undergo a process known as somatic recombination during B-cell development, where variable, diversity, and joining gene segments are randomly rearranged. This genetic mechanism allows the immune system to produce a vast repertoire of antibodies, each capable of recognizing a different antigen. Welcome to Module 2 of our comprehensive lecture series on immunology and immunochemistry. In the previous module, we explored the foundations of immunobiology, the roles of antigens and antibodies, and an overview of the immune system's components and mechanisms. Now, we will delve into more advanced concepts, including antibody structure and function, antigen-antibody interactions, detection techniques, hypersensitivity reactions, the major histocompatibility complex, and immune-related disorders such as autoimmunity and immunodeficiencies. This module is essential for understanding not only the mechanisms of immune responses but also the tools and techniques used in immunochemical studies and clinical immunology. Section 1: Antibody structure and function. Antibodies, or immunoglobulins, are Y-shaped glycoproteins produced by B cells in response to specific antigens. They play a critical role in identifying and neutralizing foreign objects like bacteria and viruses. Classification of antibodies. There are 5 main classes of immunoglobulins: IgG, IgA, IgM, IgA, and IgD. Each class differs in structure, function, and distribution. IgG is the most abundant in circulation and provides long-term immunity after infection or vaccination. IgA is found in mucosal areas such as the gastrointestinal and respiratory tracts. IgM is the first antibody produced during an immune response. IgA is involved in allergic reactions. IgD is primarily found on the surface of immature B cells. Polyspecific and monoclonal antibodies. Polyspecific antibodies recognize multiple antigens, which is useful in early immune responses. Monoclonal antibodies, by contrast, are identical and specific to a single antigen. These are important in diagnostic tests and therapies due to their specificity. Section 2: Lymphocyte isolation and function. Lymphocytes include B cells, T cells, and natural killer cells. Their roles vary but are collectively essential to adaptive and innate immunity. B cells are responsible for producing antibodies. T helper cells assist in activating B cells and other immune cells. Cytotoxic T cells kill infected or cancerous cells. Natural killer cells target virus-infected and tumor cells. Isolation techniques. Lymphocytes can be isolated from blood using density gradient centrifugation, magnetic bead separation, or flow cytometry. These techniques are vital in immunological research and diagnostics. Section 3: Antigen-antibody interactions. The interaction between an antigen and its specific antibody is a fundamental concept in immunochemistry. Characteristics. These interactions are non-covalent, involving hydrogen bonds, ionic bonds, and van der Waals forces. Specificity and affinity are key properties of these interactions. Precipitation and lattice theory. When soluble antigens and antibodies react in optimal proportions, they form insoluble complexes that precipitate. The lattice theory explains how these complexes form a lattice-like structure at the zone of equivalence. Agglutination and hemagglutination. Agglutination refers to the clumping of particles, while hemagglutination involves red blood cells. These reactions are the basis for blood typing and many diagnostic tests. Section 4: Detection of antigen-antibody reactions. Several techniques are used to detect and quantify antigen-antibody interactions. Immunodiffusion. This method involved the diffusion of antigens and antibodies in a gel, forming a line of precipitation where they meet in optimal proportions. Immunoelectrophoresis. A combination of electrophoresis and immunodiffusion, this technique separates antigens by electric charge and detects them using specific antibodies. Fluorescence quenching. This involves labeling antibodies or antigens with fluorescent tags. When the antigen binds, fluorescence is quenched or altered, indicating a reaction. Radioimmunoassay. This sensitive method uses radioactively labeled antigens or antibodies to measure minute concentrations of substances. ELISA. The enzyme-linked immunosorbent assay uses enzymes attached to antibodies to produce a measurable color change upon binding to an antigen. It is widely used in diagnostics, including for HIV and COVID-19. Fluorescent antibody techniques. These involve tagging antibodies with fluorescent dyes, allowing visualization under a fluorescence microscope. Direct and indirect methods are used depending on the application. Section 5. Hypersensitivity and allergy. Hypersensitivity is an exaggerated immune response to a harmless antigen, leading to tissue damage. There are 4 types. Type 1. Immediate hypersensitivity, e.g., hay fever, asthma. Type 2. Cytotoxic reactions, e.g., hemolytic anemia. Type 3. Immune complex mediated, e.g., lupus. Type 4. Delayed type hypersensitivity, e.g., contact dermatitis. Allergy is a type A hypersensitivity reaction. Common allergens include pollen, dust, and food proteins. Symptoms range from mild irritation to life-threatening anaphylaxis. Section 6. Major histocompatibility complex, MHC. The MHC is a group of genes coding for proteins on the surface of cells that help the immune system recognize foreign substances. MHC classes. Class 1 MHC molecules are found on all nucleated cells and present antigens to cytotoxic T cells. Class 2 MHC molecules are found on antigen-presenting cells like dendritic cells and present antigens to helper T cells. MHC restriction. T cells can recognize antigens only when they are presented with the appropriate MHC molecule. This restriction ensures that the immune system targets the correct cells. Cytokines. Cytokines are signaling proteins released by cells that regulate immunity, inflammation, and hematopoiesis. Examples include interleukins, interferons, and tumor necrosis factors. Section 7. The complement system. The complement system consists of plasma proteins that enhance the immune response. When activated, they promote inflammation, attract phagocytes, facilitate cell lysis. There are 3 activation pathways: classical, alternative, and lectin. Each leads to the formation of a membrane attack complex that can destroy pathogens. Section 8. Immunologic tolerance, autoimmunity, and immunodeficiency. Immunologic tolerance. This is the immune system's ability to recognize self-antigens and not attack the body's own tissues. Failure of tolerance leads to autoimmune diseases. Autoimmunity. Autoimmune diseases occur when the immune system targets self-antigens. Examples include rheumatoid arthritis, type 1 diabetes, and multiple sclerosis. Causes may include genetic predisposition, infections, and environmental factors. Molecular basis of antibody diversity. Antibody diversity arises from several mechanisms. Gene rearrangement, involving recombination of gene segments. Junctional diversity, created by the random addition or deletion of nucleotides at the gene segment junctions. Somatic hypermutation, introducing point mutations in the variable region genes. Class switch recombination, enabling a B cell to produce different classes of antibodies without changing the antigen specificity. These processes ensure that the immune system can recognize and respond to an almost infinite variety of antigens. Production, detection, and uses of monoclonal antibodies. Monoclonal antibodies are antibodies derived from a single clone of B cells and are therefore identical in structure and antigen specificity. They are produced using hybridoma technology, which involves fusing a specific B cell with a myeloma cell to create a hybrid cell that can produce large quantities of the desired antibody. These antibodies are used extensively in diagnostics, therapy, and research. In medicine, they are used to treat diseases such as cancer, autoimmune disorders, and infectious diseases. Concepts of clinical immunology and immune tolerance. Clinical immunology is the study of diseases caused by disorders of the immune system. These include immunodeficiencies, autoimmune diseases, allergies, and transplant rejection. Immune tolerance refers to the immune system's ability to recognize self-antigens and avoid attacking the body's own cells. When this tolerance is lost, autoimmune diseases such as rheumatoid arthritis and lupus may occur. Several factors affect immune responses, including genetics, age, nutrition, and the presence of infections or other diseases. Innate and acquired immunity, features, mechanisms, and determinants. Innate immunity is the first line of defense and includes physical barriers like skin, chemical barriers like stomach acid, and cellular defenses like phagocytes and natural killer cells. It provides immediate, but non-specific, protection. Acquired immunity is more specific and develops after exposure to an antigen. It involves the activation of B and T lymphocytes and leads to immunological memory, which allows for a faster and stronger response upon subsequent exposures to the same antigen. Determinants of immunity include genetic background, health status, nutritional status, environmental factors, cells and organs of the immune system. The immune system comprises several cell types and organs that work together to protect the body. Primary lymphoid organs, bone marrow and thymus, responsible for the production and maturation of lymphocytes. Secondary lymphoid organs, lymph nodes, spleen, tonsils, sites where immune responses are initiated. Key immune cells include B lymphocytes, produce antibodies. T lymphocytes, help or kill infected cells. Macrophages and dendritic cells, present antigens and activate other immune cells. Natural killer cells, destroy virus-infected and tumor cells. Evolution of the immune system. The immune system has evolved over millions of years. Invertebrates rely primarily on innate immune mechanisms, while vertebrates, including humans, have developed both innate and adaptive immune responses. This evolution has led to a sophisticated system capable of learning and remembering past infections, which forms the basis of vaccination. Conclusion. In this first module, we have covered the foundational principles of immunology and immunochemistry. We explored the nature of antigens and antibodies, the structure and classification of immunoglobulins, the different types of immunity, and the critical roles played by various immune cells and organs. We also looked into the mechanisms that generate antibody diversity and how monoclonal antibodies are produced and used. In the next module, we will delve into the advanced biochemical interactions of immunochemistry, focusing on antigen-antibody reactions, immunoassay techniques, immune system disorders, and immunologic applications in diseases and transplantation. The role of nanoparticles in oil spill management. Recent advancements in nanotechnology have opened new frontiers in environmental protection, especially in carbon oil spills. Nanoparticles possess unique physical and chemical properties that make them ideal for environmental remediation. 1. Absorption and degradation. Certain nanoparticles can absorb and break down hydrocarbons, making cleanup more effective. 2. Surface modification. Nanoparticles can be engineered to interact specifically with oil, enabling more precise targeting of contaminants. 3. Oil-water separation. Magnetic nanoparticles can be used to separate oil from water, allowing for easier recovery and treatment. 4. Soil and water remediation. Nanomaterials can be applied to contaminated sites to bind and neutralize pollutants, restoring environmental quality. Despite their promise, the application of nanoparticles must be carefully monitored to avoid unintended ecological effects. Ongoing research is essential to maximize benefits while minimizing risks. Environmental parameters in health and safety. Monitoring environmental parameters is a key component of HSE. These parameters include 1. Physicochemical parameters. These include temperature, pH, dissolved oxygen, chemical oxygen demand, COD, biological oxygen demand, BOD, and turbidity. Monitoring this helps assess the quality of water and air in and around project sites. 2. Human interest parameters. These refer to noise levels, odor, visual aesthetics, and accessibility of resources. They reflect the perception and experience of individuals affected by industrial activities. 3. Ecological parameters. These involve the study of flora and fauna, biodiversity indices, and habitat quality. Ecological monitoring helps identify changes caused by industrial operations and aids in conservation planning. Regular assessment of these parameters enables the identification of risks, implementation of control measures, and continuous improvement in environmental performance. Environmental Impact Assessment, EIA definitions and objectives. An environmental impact assessment, EIA, is a systematic process used to evaluate the potential environmental effects of a proposed project before it begins. The main objectives are to identify, predict, and evaluate the potential environmental impacts. To propose measures to prevent, reduce, or mitigate adverse effects. To provide decision-makers with relevant information to approve or modify projects. EIAs are crucial in balancing development needs with environmental sustainability. They ensure that the long-term impacts of projects are considered and addressed proactively. EIA Directive and projects requiring EIA. The EIA Directive, adopted in various jurisdictions, provides a legal framework for conducting environmental assessments. It outlines types of projects requiring EIA. These include oil and gas exploration, dam construction, mining, industrial facilities, and large-scale agriculture. Stages of EIA: screening, scoping, impact analysis, mitigation planning, reporting, and review. Each step is important in ensuring that environmental concerns are integrated into project planning and decision-making. Evolution of EIA. EIA practices have evolved significantly over the decades. Initial focus: early EIAs concentrated mainly on biophysical impacts. Broadening scope, they now include social, economic, health, and cultural dimensions. Increased public participation, communities and stakeholders now play a more prominent role in the EIA process. Integration with sustainability goals, modern EIAs consider long-term sustainability, climate change, and ecosystem services. This evolution has enhanced the relevance and effectiveness of EIAs in promoting responsible development. Types of EIA and core values. EIAs can take different forms depending on the nature of the project. 1. Preliminary EIA, conducted at the conceptual stage to identify significant issues. 2. Comprehensive EIA, a detailed study including extensive baseline data and impact modeling. 3. Strategic Environmental Assessment, SEA, focuses on policies, plans, and programs rather than individual projects. 4. Cumulative Impact Assessment, evaluates the combined effects of multiple activities over time. Core values guiding EIAs include transparency, scientific integrity, public involvement, and a commitment to sustainable outcomes. EIA process in Nigeria. The Nigerian EIA process is governed by the Federal Ministry of Environment and is guided by the EIA Act. The process includes the following steps. 1. Screening, determining whether a project requires an EIA. 2. Scoping, identifying key environmental issues and stakeholders. 3. Impact assessment, collecting and analyzing data to predict potential impacts. 4. Public consultation, engaging with affected communities and stakeholders. 5. Review and decision, the environmental authority reviews the EIA report and makes a decision. 6. Monitoring and compliance, ensuring that mitigation measures are implemented and monitoring continues during project execution. The Nigerian EIA framework aims to align industrial growth with environmental responsibility, ensuring that future generations inherit a safe and healthy environment. Conclusion: quality health, safety, and environment studies are central to modern industrial operations. By adopting good housekeeping practices, ensuring effective communication, managing waste responsibly, adhering to regulatory guidelines, and conducting environmental impact assessments, organizations can create a safer workplace and a more sustainable environment. The integration of science and technology, such as the use of nanoparticles and advanced treatment methods, continues to enhance the capacity for environmental protection and occupational health. In a rapidly changing world, maintaining strong HSE standards is not just a regulatory requirement, but a strategic imperative for long-term success and societal well-being. Immunodeficiency. This refers to the inability of the immune system to mount an adequate response. It can be primary, genetic disorders like severe combined immunodeficiency, SCID. Secondary, acquired conditions like HIV, AIDS or chemotherapy-induced suppression. Section 9. Transplantation and Immunology of Parasitic Diseases. Transplantation immunology. Transplants often trigger immune responses due to MHC mismatches. Rejection can be hyperacute, minutes to hours; acute, days to weeks; chronic, months to years. Immunosuppressive drugs are used to reduce the risk of rejection. Immunology of parasitic diseases. Parasitic infections trigger complex immune responses. Parasites often evade detection by changing surface antigens or suppressing immune function. Understanding these mechanisms is crucial for vaccine development and treatment strategies. Conclusion: this module has provided a comprehensive overview of advanced topics in immunology and immunochemistry, building upon the foundational principles discussed in Module 1. We have explored how antibodies function and interact with antigens, the laboratory techniques for studying these interactions, the nature of immune hypersensitivity, and disorders like autoimmunity and immunodeficiency. We also touched on the complexities of transplantation and parasitic disease immunology. A solid grasp of these concepts is essential for students, researchers, and healthcare professionals alike. Immunology is a dynamic and ever-evolving field, and staying informed about its principles and applications is crucial for tackling present and future challenges in health and medicine. Thank you for listening to this lecture. We hope it has enriched your understanding and appreciation of the intricate workings of the immune system. Molecular basis of antibody diversity. Antibody diversity arises from several mechanisms. Gene rearrangement, involving recombination of gene segments. Junctional diversity, created by the random addition or deletion of nucleotides at the gene segment junctions. Somatic hypermutation, introducing point mutations in the variable region genes. Class switch recombination, enabling a B cell to produce different classes of antibodies without changing the antigen specificity. These processes ensure that the immune system can recognize and respond to an almost infinite variety of antigens. Production, detection, and uses of monoclonal antibodies. Monoclonal antibodies are antibodies derived from a single clone of B cells and are therefore identical in structure and antigen specificity. They are produced using hybridoma technology, which involves fusing a specific B cell with a myeloma cell to create a hybrid cell that can produce large quantities of the desired antibody. These antibodies are used extensively in diagnostics, therapy, and research. In medicine, they are used to treat diseases such as cancer, autoimmune disorders, and infectious diseases. Concepts of clinical immunology and immune tolerance. Clinical immunology is the study of diseases caused by disorders of the immune system. These include immunodeficiencies, autoimmune diseases, allergies, and transplant rejection. Immune tolerance refers to the immune system's ability to recognize self-antigens and avoid attacking the body's own cells. When this tolerance is lost, autoimmune diseases such as rheumatoid arthritis and lupus may occur. Several factors affect immune responses, including genetics, age, nutrition, and the presence of infections or other diseases. Innate and acquired immunity, features, mechanisms, and determinants. Innate immunity is the first line of defense and includes physical barriers like skin, chemical barriers like stomach acid, and cellular defenses like phagocytes and natural killer cells. It provides immediate, but non-specific, protection. Acquired immunity is more specific and develops after exposure to an antigen. It involves the activation of B and T lymphocytes and leads to immunological memory, which allows for a faster and stronger response upon subsequent exposures to the same antigen. Determinants of immunity include genetic background, health status, nutritional status, environmental factors, cells and organs of the immune system. The immune system comprises several cell types and organs that work together to protect the body. Primary lymphoid organs, bone marrow and thymus, responsible for the production and maturation of lymphocytes. Secondary lymphoid organs, lymph nodes, spleen, tonsils, sites where immune responses are initiated. Key immune cells include B lymphocytes, produce antibodies. T lymphocytes, help or kill infected cells. Macrophages and dendritic cells, present antigens and activate other immune cells. Natural killer cells, destroy virus-infected and tumor cells. Evolution of the immune system. The immune system has evolved over millions of years. Invertebrates rely primarily on innate immune mechanisms, while vertebrates, including humans, have developed both innate and adaptive immune responses. This evolution has led to a sophisticated system capable of learning and remembering past infections, which forms the basis of vaccination. Conclusion. In this first module, we have covered the foundational principles of immunology and immunochemistry. We explored the nature of antigens and antibodies, the structure and classification of immunoglobulins, the different types of immunity, and the critical roles played by various immune cells and organs. We also looked into the mechanisms that generate antibody diversity and how monoclonal antibodies are produced and used. In the next module, we will delve into the advanced biochemical interactions of immunochemistry, focusing on antigen-antibody reactions, immunoassay techniques, immune system disorders, and immunologic applications in diseases and transplantation.